In this video, we're going to be going over cell division, which is extremely important from a physiological process and understanding how cells divide, differentiate. It's also important from a clinical perspective because it's estimated that nearly half of all Americans are going to develop cancer during their lifetime. And we're going to see that cancer is just a malfunction of the normal cell division, where the cells divide uncontrollably. In just a past second, millions of your cells have divided in two. It doesn't mean it only takes a second to divide in two, but these cells are undergoing a process which may take hours to a day or two, and then they're ready to divide. In this video series, we're going to be going over the types of cell division. We're going to go over chromosome number. We're going to go over the cell cycle with a focus on mitosis. We'll talk about meiosis, but we're going to cover meiosis in more detail when we get to the reproductive system. Here we're going to focus primarily on mitosis and talk about the physiology of cancer cells. Cell division is a process where cells reproduce themselves. You have stem cells, for example, and they can continue to divide while their daughter cells become specialized through a process called differentiation. And you want to make sure you star differentiation. And basically what happens is that these cells, as they differentiate, the genes that the cell doesn't need to have activated for their normal function are shut down. And the genes that the cells need to do their job are used. And think about this. Every cell in your body, except your sex cells, have the same genetic material, the same chromosomes, the same genes, but they don't do the same things. For example, when you look at in your eyes, you have your iris. Well, those cells that make up your iris are expressing genes functioning differently than, say, your liver cells are, which are using different genes for their function. But they all have the same genetic material. So the iris cell has genes shut down that it doesn't need to have expressed. And then your liver cells have genes that are shut down. Well, what do we call the process of shutting down these genes that that cell doesn't need and then activating those genes that the cell does need? We call that differentiation, which allows cells to mature and perform specific functions. When you look at the functions of cell division, this is to replace diseased and damaged cells, growth of the organism, and production of new organisms. There are two types of cell division: somatic cell division and reproductive cell division. Let's test our knowledge. Go ahead and pause the video and then do the fill-in here and then answer these two questions. Here are the answers. When you look at somatic cell division, this is where the cell undergoes mitosis, which is a type of cell division that's going to result in two daughter cells that are genetically identical. They have the same number and type of chromosomes as the parent cell. The cell divides in a process of cytoplasmic division called cytokinesis. And what we're going to see is that the chromosomes are going to get replicated. Those replicated chromosomes are going to separate from each other and move to opposite poles. And this process is referred to as mitosis, is the movement of these chromosomes to opposite poles. And here you have the chromosomes here and the chromosomes here after they've been replicated. Well, now the cell has to divide in two. The cytoplasm has to split, and we call cytoplasmic division cytokinesis. In reproductive cell division, the cells are going to undergo a process of meiosis that produces gametes, such as eggs and sperm, that are genetically different than the parent cell. And the egg and sperm contain half the number of chromosomes as the parent cell. So we're going to see that your somatic cells each have 46 chromosomes, but the egg and sperm have 23 chromosomes. They have half the number of chromosomes. Meiosis occurs in the ovaries of females and the testes of males. The function: the ovaries are going to produce eggs that have half the number of chromosomes. The testes are going to produce sperm that have half the number of chromosomes. The creation of offspring by fertilization of that egg by a sperm brings the chromosome number back to 46. 23 in the egg, 23 in the sperm come together to give you 46 chromosomes. Here's the sperm uniting with the egg, and that's going to implant. That fertilized egg, which we refer to as a zygote, has 46 chromosomes. Let's go over some background information on chromosomes. Each somatic cell contains 46 chromosomes. You can also say 23 pairs of chromosomes. And if you do this on a quiz or exam and you use 23, make sure you put pairs after it. If you just put 23 and we're talking about a somatic cell, it'd be incorrect. It's 23 pairs of chromosomes. Well, one member of each pair came from your mother, and the other member of a pair came from your father. Somatic cells contain the diploid number of chromosomes. You want to star that because they contain both sets of chromosomes. They have two sets. of 23 chromosomes to equal 46 total chromosomes, and refer to organisms that have 2 sets of chromosomes as the diploid organism, or the diploid number of chromosomes. Now, of these 23 pairs, 1 pair are the sex chromosomes, which consist of 2 X chromosomes if the person is a biological female, or an X and Y chromosome if the person is a biological male. The remaining 22 pairs of chromosomes are called the autosomes. Sex cells, such as sperm and eggs, contain 23 individual chromosomes, or 1 set, which is called the haploid number. Make sure you star that. Or half the number of chromosomes. It only has 1 set of chromosomes. It has half the number of chromosomes. So think of haploid as half. Well, the union of a sperm and egg during fertilization is going to restore that chromosome number to 46, or 23 pairs. A karyotype is a test to identify and evaluate the size, the shape, and the number of chromosomes in a body cell. A normal karyotype has 22 pairs of autosomes and 1 pair of sex chromosomes to give you 23 total pairs. And the genome is the genetic content of a cell or organism. Shown here is a typical karyotype. What they've done, they stain and they take pictures of the chromosomes. And then they cut out those chromosomes. They're not normally right next to each other like this. You can see these little lines here where they cut out the chromosome and then they match them up by their size and shape. And that gives you a karyotype. So chromosome number 1 here, we have 2 of those. We have 1 that came from your mother, 1 that came from your father. Chromosome number 2, 1 from your mother, 1 from your father. And they match these up during a karyotype. This is going to allow you to see if there's any physical chromosomal abnormalities, as well as if there's an abnormal chromosome number. For example, if you were the technician doing the karyotype and you found 3 of chromosome 21, what would that condition be called? Well, it's called trisomy 21 or Down syndrome. So here's an egg which has 23 chromosomes, or the haploid number, uniting with a sperm which has 23 chromosomes, or the haploid number also. They unite together, and that's going to give you a diploid zygote. It's a fertilized egg. Let's test our knowledge. Go ahead and pause your video and select the term that makes a sentence correct. Here are the correct answers. Pause your video and select the term that makes a sentence correct. Here are the answers. Pause the video, select the term that makes a sentence correct. Here are the answers. In the next video, we're going to focus on the cell cycle.